In this video, I'm going to talk about gluconeogenesis, an important process for maintaining our blood sugar at the appropriate level. It also ties in really closely with glycolysis and glycogen. So if you haven't seen my videos on that, it might be worth checking them out before diving into this one. But let's go ahead. So one of the things that our physiology cares a lot about is maintaining our blood glucose concentration into a fairly narrow range. This is in response to things both acute, so we eat or we don't eat. For brief periods of time, our blood sugar fluctuates, and we have compensatory mechanisms to keep that blood sugar within a narrow range. This also occurs over long-term things like starvation. So if you don't eat for a very long time, your blood glucose concentration remains relatively constant because of homeostatic mechanisms. I've likened this in the past to a biological cruise control, right? When you set your cruise control in the car, measures your speed and adjusts your throttle to keep that speed the same. We see the same thing with blood glucose concentrations, where if your blood glucose concentration starts to change, we have mechanisms that either add or take away glucose to keep it at a specific concentration. And one of the things we've talked about in the past is that if we have a muscle cell that's consuming, or muscle cells, I guess, that are consuming lots of glucose, they're going to be importing glucose from the blood. That's going to trigger the release of glucagon by the pancreas, which is then going to trigger liver cells to start breaking down their own glycogen to drop it into the bloodstream. But there's another parallel process that happens, which is actually gluconeogenesis. Liver cells are going to start synthesizing glucose, and that's what we're going to be talking about in this video. To situate this in our transit map analogy we've used in the past, what we've talked about is glycolysis, which is basically going down from glucose towards pyruvate. What we're going to be talking about today is the exact opposite, going from pyruvate up to glycolysis. And if we situate ourselves on the sort of descriptive map of things, what we're basically, we're going to throw things. What we're talking about this here is gluconeogenesis, the reverse line of glycolysis, making glucose from pyruvate. All right, so what are we going to talk about? First, what is gluconeogenesis? And gluconeogenesis is simply the reverse of glycolysis. So in glycolysis, we take a glucose, we make some pyruvates, we spit out some ATPs and some NADHs. Gluconeogenesis is the exact reverse at the level of the glucose and the pyruvate. In many of the molecules, it's just we're going to add in some extra ATP consumption along the way. It occurs primarily in the liver, as well as a few other organs that are known as gluconeogenic tissues or gluconeogenic organs. But the liver is the main focus because this is the place where it's doing gluconeogenesis to maintain our blood sugar at a particular level, just like it's releasing breaking down glycogen to release glucose. It starts making glucose, so that's all happening in the liver. So everything I'm going to be talking about is for sort of a liver gluconeogenic process. Basically, gluconeogenesis is a heck of a lot like glycolysis. Most of its steps are in reverse, so I'm actually showing here glycolysis, the entire process of going from glucose to pyruvate by 10 steps. Gluconeogenesis adds a set of steps that reverse a few of them, but overall, like if I'm drawing a circle, this is all identical between the pathways as well as this. So 7 of the 10 reactions are identical between gluconeogenesis and glycolysis because they're basically reversible. Remember, we've actually talked about that. We did some calculations on the energies of these, but basically these reactions are essentially at equilibrium. The delta G for these reactions is about 0 in cells. So it doesn't matter. They will flow forward or backwards, no problem. But these steps here are not the same, and it's because those steps are not reversible. And so actually I'm going to, I deleted them. I deleted the reverse step. So let's erase our beautiful art here so you can really appreciate, oh my goodness. You can really appreciate what I was trying to draw graphically there, sorry. But that what gluconeogenesis does is it does glycolysis with a sort of bypass around those reactions. Okay, I don't know if that was worth all the erasing, but we did it anyway. So which reaction does it bypass and why? What it does is it bypasses reactions where the forward reaction for glycolysis has a delta G much less than 0. And so for example, the first reaction, glucose going to glucose 6-phosphate, that is involves an ATP, right? We're taking ATP and we're taking the phosphate off of that ADP to put it onto the glucose. That's very favorable. Can't run that backwards, so gluconeogenesis runs a different reaction in reverse. In this case, it's just going to chop the phosphate off. And what's interesting is it ends up making both reactions are simultaneously favorable. So here I'm showing the core thing that we're talking about here in the glycolysis direction. A glucose turns into a glucose 6-phosphate. The way that this happens in glycolysis is an ATP takes its phosphate, sticks it onto the 6th position, leaving a glucose, or sorry, I drew it onto the 1 position, onto the 6th position, rather. And the ATP loses its phosphate, right? It goes to ADP. This has an energy, standard state energy of minus 16.7 kilojoules per mole. So it's very favorable to do this because we're hydrolyzing ATP. Gluconeogenesis actually runs a reaction that is also quite favorable, 13.8 kilojoules per mole. And that is just taking this and chopping it off, right? So if you just chop off the phosphate, it's energetically favorable. And incidentally, these two reactions add up. So this is, well, never mind. I won't go to that. So great. So this is energetically favorable to go backwards, energetically favorable to go forwards. As far as the glucose is concerned, it's an exact reverse, glucose to glucose 6-phosphate, glucose 6-phosphate to glucose, but the actual reactions are different. You're using either an ATP or you're just chopping off the phosphate. Turns out, though, that if you think about it, all of these backward steps are energetically expensive. So if we do this, if we start, let's look at the top here. If we start with ATP and we make an ADP to make G6-phosphate, and then we go backwards, we lose that, right? So we're basically losing our ATP when we go backwards. The same thing happens at the phosphofructokinase step, which is also reversed, where you lose the phosphate, so that's energetically expensive. But the most obviously energetically expensive part is down here when we go from pyruvate, the endpoint of glycolysis, to phosphoenolpyruvate, the second from endpoint in glycolysis. When glycolysis runs, it's just, oh, look, this is so energetically favorable that we actually get an ATP out. So to go backwards, you have to do a lot of work. And it turns out the work involves the investment of an ATP to take a pyruvate and convert it into oxaloacetate, and then taking the oxaloacetate and converting it back to phosphoenolpyruvate using a GTP, which is the same energy as an ATP, just has a guanine base rather than an adenine base. So this is energetically costly. Now, I'm not going to actually go into the details of why we do that particular mechanism, because actually we have to develop some more ideas with the mitochondria and other things. So we'll come back to that later. But for the moment, what we can say is that basically gluconeogenesis invests energy to make glucose. It takes basically 6 ATP equivalents to go from 2 pyruvates to glucose, right? You've got your 2 ATPs that you would have had to invest anyway to go backwards stoichiometrically. And then you also have the 2 ATPs that you invest and the 2 GTPs you invest over the entire process. Okay. What are the inputs to gluconeogenesis? So it's this expensive process that we employ when we need to make glucose to stick into our bloodstream. What are the inputs? There's a variety of inputs. We already mentioned one in the video about fermentation, but recall that if you have a muscle cell that is operating with anaerobic fermentation or lactic acid fermentation, it's putting lactate out into the bloodstream, which I'm just going to call L. That can get imported into liver cells, where it gets converted into pyruvate, and then back into glucose by the process of gluconeogenesis, and then re-exported into the bloodstream, right? So this is the Cori cycle. This is one source of glucose for, or one source of feedstock for gluconeogenesis. Another feed source is if there's a lot of fat around, but not very much else. The glycerol backbone of triglycerides, which are the way we move around fats, can also enter this pathway. Instead of entering all the way down at pyruvate, they actually enter as dihydroxyacetone phosphate. And maybe you remember that because that is one of the glycolytic intermediates. So dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are the result of the split when we go from the 6-carbon fructose 1,6-bisphosphate to the 2,3-carbon subunits. Dihydroxyacetone phosphate is one of those subunits. So it's not coming all the way down at pyruvate, but it is entering the top part of the gluconeogenic pathway, glycerols from fats. The last and probably the most important source of fuel for this is actually amino acids. So basically every single amino acid, except for leucine and lysine, can be converted to pyruvate or oxaloacetate. And so pyruvate is the end of glycolysis, right? And so pyruvate can go to oxaloacetate and then pyruvate. A bunch of different amino acids can come straight in. When they are catabolized into pyruvate. Another subset get catabolized into oxaloacetate, so they're kind of halfway there already. But what this means is actually, this is one of the reasons that it's really important if you're on a super low carb diet and basically eating no carbohydrates, you also need to have a high protein diet. And that's not just to try to build muscle, but it's also because if you don't have enough amino acids around, your body's going to start breaking down muscle because it needs to keep your blood sugar up. And where is it going to get the material to do that? It's going to do it by breaking down proteins, whereas most of your free protein in your body, it's in your muscles, right? So that's why things like the keto diets and everything, it's a mix of low sugar and high protein so that you can keep your blood sugar up even though you're not eating any sugar. Okay, last question, and I was going to answer in this video, is how's gluconeogenesis regulated? It felt like a complicated topic on its own, so I'm actually going to table that and talk about that in the next video. So we're actually done with our high-level survey of gluconeogenesis. Quickly to describe it though, or to summarize it, gluconeogenesis synthesizes glucose in the liver for export into the bloodstream. It takes feedstocks, which through first approximation, it's mostly amino acids, but also lactate and glycerol, and it can run glycolysis in reverse to generate glucose. And it bypasses favorable reactions of glycolysis with alternate reactions that take energy. So it's energetically expensive, but at the end of the day, you get out this glucose molecule that you need to keep your blood glucose high. All right, so that is what I have on glycolysis. We've got, the next video is going to be about the regulation of glycolysis. Too many G words. That was our video about gluconeogenesis. And we are going to be talking about the regulation of gluconeogenesis and glycolysis together in the next video. So hope to see you there. And until then, thanks for watching.